Good afternoon, everybody. Thank you, Russ, for the introduction. I think I'm just going to repeat Russ in the next two slides. Thank you for coming here today. The title of my presentation is The Development and Regulation of Next Generation Precision Medicine. Like Russ mentioned, my name is Anuradha Ramamoorthy. I'm a reviewer within the Genomics and Targeted Therapy group at FDA. And I'm joined today by two of my colleagues, Dr. Mike Pakanowski, who is the Associate Director for the Genomics and Targeted Therapy Group, and Dr. Oluseyi Adeniyi, who's a Commissioner's Fellow in our group. Can you hear me clearly? Or am I
You need to speak up.
Okay.
I don't think the microphone works.
There isn't a microphone, so I'm going to scream. So FDA's genomics and precision medicine universe is fairly large. There are a number of centers that are involved in various activities that's central to their mission. For example, there's Center for Drugs, Center for Devices, Center for Biologics, etc. The Genomics and Targeted Therapy Group sits within the Office of Clinical Pharmacology under the umbrella of Office of Translational Sciences. Does this work? Or this is, there. Okay, so under the umbrella of Office of Translational Sciences, which is within the Center for Drugs. And it's with this perspective that I'm going to be talking today. In terms of agenda, the first part of my talk will focus on the development of precision medicine, what's approved and general thoughts about what's in the pipeline, as well as regulatory framework that support the development and approval of precision medicine. So for the first part of the talk, which is the development of precision medicine, so what is precision medicine? I'm sure everybody has seen this picture, probably used quite a lot since President Obama announced the Precision Medicine Initiative in his State of Union talk last year. For the purposes of PMI, precision medicine is treatment tailored to an individual's gene, environment, and lifestyle. And within that, I'm going to specifically talk about the genetic aspects of this continuum. So for the purposes of my talk, precision medicine is a drug or biologic that is intended for use with a specific biomarker, either for selecting patients to receive a particular therapy or to select doses for those patients or to monitor response. And these biomarkers could be genomic, proteomic, or other specific kind of biomarkers, and they can be prognostic, predictive, or response biomarkers or related values. And in general, they are tied to the pharmacology of the disease, of the therapeutic product, that is. And because of that, pharmacogenomics or pharmacogenetics can have great impact. Pharmacogenetics can affect a therapeutic product's exposure or response. With respect to exposure, it can affect the pharmacokinetics by affecting the ADME process, either metabolism or even transportation of the therapeutic product. In terms of affecting response, it could be through the efficacy, which is related to the drug target or the target pathway, or by being a susceptibility or prognostic biomarker. And in terms of safety, this could be modulated because of the intended target or unintended target, which is the off target, or through immunologic or idiosyncratic processes. But when we use the term pharmacogenetics or genomics, it typically brings to mind the bimodal distribution of metabolites based on enzyme activity, with classic examples of 2D6, 2C9, 2C19, and a genotype or phenotype-based drug dosing. But pharmacogenetics and genomics is just not ADME or ADMET genes. It's more than that. When we look at biomarkers and genetic factors that are included in product labeling, as of May of this year, there were about 198 gene-drug pairs, of which still a majority of 43% were tied to metabolism and transportation, about a third were tied to the target or the pathway, and about a quarter to the immunological or other safety mechanisms. And nearly 50% were considered actionable, that is, the genotype or phenotype is listed in the product labeling, either in boxed warning or dosage and administration or indication and usage or warning and precaution sections of the labeling. And the biomarker was tied to explicit recommendation related to dosage or usage of that therapeutic product. And here I want to acknowledge work done by Stanford University folks, particularly Dr. Russ Altman and his team. If I have any question on some of these pharmacogenes or pathways, the first place I go to is PharmGKB. So if you're not familiar with PharmGKB, I would say go check it out. And moving back to what's in the labeling with respect to genetic biomarkers, initially, many years ago, when you saw a product labeling and a biomarker was listed in it, it was most likely added in the post-approval space. That is, a drug was approved and more knowledge was accumulated about the role of the biomarker, and then it was added to the product labeling. For example, DYPD, in case of capecitabine and fluorouracil, was added in the post-market space. Over the past many years, this has changed, that we have enough information about the biomarker in the pre-approval space that it's added to the product labeling much earlier on at the time of approval. One of the example is olaparib, which is a PARP inhibitor that's approved for BRCA, for ovarian cancer. Information about BRCA mutation was available early during development, that the biomarker was studied extensively during the clinical trials, and the biomarker was added to the product labeling at the time of approval. And the drug was co-approved with a companion diagnostic. And there have been many examples of companion diagnostics that have been co-approved along with the therapeutic product. Olaparib is one of the examples. The companion diagnostic that was co-approved helps to detect patients who had deleterious or suspected deleterious germline BRCA mutation. And there are other examples as well. For example, first generation and second generation EGFR tyrosine kinase inhibitors like afatinib, gefitinib, and erlotinib all have companion diagnostics that help to identify patient tumors that have specific EGFR mutations, like EGFR exon 19 deletions or EGFR exon 21 L858R mutations. And there are a number of other examples as well. The one thing I wanted to mention here was a recent trend in drug development are the complementary diagnostics. I'll be talking about complementary diagnostics pretty briefly later during my talk, but I just wanted to mention here that there have been a couple of those approved, mostly in the oncology space, in the PD-L1 space. So I hope these examples have convinced you that it is possible to co-develop a drug and a diagnostic, and that precision medicine is really upon us. About 1 in 5 of the novel new drugs that have been approved in the past 2 years are considered to be precision medicine. And that makes sense given that when we look at top 10 drugs that have been approved in the US, about 4 to 25% of the patients who receive these medications actually receive benefit from these drugs. So it makes sense to move from this imprecise medicine more towards precision medicine. And taking into account the variability also will help to address another challenge that's faced by the drug development industry, and that of the very high attrition rate. The average industry success rate for a new drug to hit the market is thought to be about 4%. So if you had roughly 14.6 compounds in the preclinical stage, that results in the launch of one new molecule. But that also varies across therapeutic areas. So for example, if you were in the HCV space, it takes about 30 new compounds in the preclinical space. If you were in Alzheimer's, it takes 119 compounds to get to one successful launch. And that's because of the very high attrition rate along the entire length of this funnel. And when this happens in phase 2 and phase 3, these become really expensive failures. About 50% to 60% of failures in phase 2 and 3 are thought to be because of efficacy concern, and about 20% are thought to be because of safety concerns. So if we are able to address those failures, that would save a lot of resource. And this is where having a good understanding of human genetics and having a good understanding of the druggable genome will help to address the attrition rate. About 3,000 genes were thought to be druggable, and this was in 2002. Since then, there's been a lot of technological revolution, not just next generation sequencing, but also the bioinformatics support that goes with the technological revolution. And a recent study that was published in July of this year suggested there are about 4,000 druggable genes, and this adds nearly 2,400 new targets to the already existing studies. And these studies are acute reminders of targets not being pursued, that is, the white spots in the pharmaceutical pipeline, if you will. And having a good understanding of what makes a target will help to bring novel therapies to the patients, be it small molecule or biologics targeting the protein, or even other therapeutic modalities like antisense oligos targeting nucleic acid sequences. The ultimate hope is that these innovations would help to transform patient care, especially for diseases with high unmet medical need. And there's evidence that's beginning to accumulate that genetics is important and should be considered when making key decisions related to drug development programs. This was a study published by GSK folks looking at drugs that have been already approved and the targets that are associated with it. And what they found was if the target had a genetic evidence that supports its role in the disease or the indication that's being sought, it's twice as likely to succeed when compared to a target that didn't have a genetic basis. I think to a lot of us that sounds like a no-brainer, but that's not really how it has worked so far. And this has been replicated in many shapes and forms by other studies. One of this is a study done by AstraZeneca looking at their own pipeline and looking at compounds in phase 2. And what they found was if a target had a human genetic linkage to the disease indication, or if the compound had an efficacy biomarker, the probability of success was much higher when compared to not having a genetic link of the target to the disease or not having an efficacy biomarker. Another study that was published by Bio organization looked at having biomarker-based testing as a part of inclusion criteria for the clinical trial, and said that the chances of success is much higher if there was a biomarker-based testing. And in fact, the chances went up by 1 in 10 to about 1 in 4. And genetics is, because of all of these reasons, genetics and biomarkers are now being incorporated into drug development framework. A good example comes from AstraZeneca and Merck. AstraZeneca published this 5R model, which is right target, right tissue, right safety, right patient, and right commercial potential. And Merck published their translational medicine guide, which had 3 steps, which is trust in target, trust in therapeutic window, trust in patient population. So whether it's the 5R model or the 3-step 3T model, you can see where genetics and biomarkers can fit into every step, be it exposure, identifying the exposure differences or identifying response, or selecting patients and including even thoughts about co-development strategies. So, typically, when you're thinking about targeted development approaches, ideally, these targeted approaches are planned from the earliest stages. This can happen when the predictive prognostic biomarkers are known and there are clear genetic differences in the target. And typically, by the end of phase 1 or phase 2, the target population is very well defined. And discovery can be based on small trials fit to identify large effect size. Preliminary assessment of predictive value is possible, and uncertainties can be reduced during subsequent randomized controlled trials. Exceptionally, biomarkers are identified in the later phases or post-approval, and this can happen when there's a compelling shift in the risk to benefit ratio. When this happens, complete high-quality data are very critical to make decision, and because of the prospective retrospective nature, this requires careful planning. And thought should be given to the nature of the biomarker that's being used, whether it's predictive, prognostic, or response biomarker. Understanding the role of the biomarker can help to stratify, enrich in the clinical trials, and ultimately these biomarkers can be used for patient selection, that is to identify patients who are likely to respond, patients who are not likely to respond, or even subset of patients who may have toxic responses. It can help to identify doses as well as monitor for response. And this is becoming possible as we gain better understanding into the molecular nature of the disease and understand what the drivers of particular disease are. If you take non-small cell lung cancer, knowing if it's being driven by EGFR or ALK or ROS is helpful to decide which path to go down. And that's also becoming possible because of the technological revolution and the big data that's being generated and knowledge being accumulated because of that. The hope is that all of these research lead into rational drug development program, rational next generation tests, and ultimately better care for the patients. And that's happening more frequently in oncology and rare diseases than the common chronic morbid diseases. It's been much slower for other diseases, and it's being much slower in translating these research to develop targeted therapeutics. One of the studies that was published recently looking at cardiovascular drug development space for the past 20 years concluded that even though there are more novel targets in the latter part of the study period, there was still a huge amount of failure, about 44% due to efficacy and about 25% due to safety concerns. And so that's still unacceptably high failure rate. This is where, in order to understand where the bottleneck is and where the white spots are, we have two ongoing projects in our group. The first one is evaluating the current pharmaceutical pipeline, especially for diseases with high public health need. The goal is to try and evaluate the extent of biomarker-based drug development, to identify the presence of pharmacogenomic liability. For example, is there racial ethnic differences, or if there are high pharmacokinetic variability or presence of outliers, or if there are target gene variation. In order to understand if these pharmacogenomic liabilities can be managed through prospective biomarker assessment or targeted drug development strategies. And this is work being done by Oluseyi Adinei, who is a Commissioner's Fellow in our group. And as a sidebar, if you have any questions about Commissioner's Fellowship Program, talk to her after this session. She'll be happy to answer your questions. I told you I was going to throw you under the bus. The second project is looking at deconstructing the late phase safety failures. Safety issues arise in the pre-market space as well as the post-market space. In the post-market space, it can lead to withdrawal of a drug, or most often leads to labeling changes. While in the pre-market space, it can lead to non-approval or delayed approval of the drug, phase 2-3 suspensions, or INDs being placed on clinical hold. The goal of this project is to deconstruct drug development practices so that we can answer questions like, can we predict and preempt safety signals through genome-based phenome mapping? Or can we mitigate some of the safety concerns through biomarker studies or targeted drug development? And this is work being done by Shala Riazi, who's an ORISE fellow in our group. That was with respect to the development of precision medicines. Switching gears, I'll talk briefly about the regulatory framework that supports the development and approval of precision medicines. FDA has been working for over a decade to drive scientific advancement to enable the development and approval of precision medicines. There are over 10-12 guidances here, I think, that support precision medicine area, including some of these are draft guidances, as well as ICH, or International Council of Harmonization guidances. I will specifically talk about 4 of these guidances briefly. There is links at the bottom of this slide. I don't know if you have access to these slides. If not, you would be able to just Google for FDA guidances and you will find some of these. The first one is the Clinical Pharmacogenomics, or pre-marketing evaluation and early phase clinical studies and recommendations for labeling. The purpose is to guide industry on evaluating the impact of genomic variation, particularly the variations in the DNA, to see how that affects the drug's PK, PD, safety, or efficacy. And it provides general principles on the pharmacogenomic studies, including study design, data collection, data analyses, etc. And the scope is limited to the early phase clinical studies. It doesn't specifically address the late phase randomized control trials, and it does not address tumor genomics specifically. The second one is the enrichment guidance. So for the purpose of this guidance, enrichment is defined as the prospective use of any patient characteristic to select a study population in which it's more likely to detect a drug effect when compared to an unselected population. And there can be 3 strategies when you're thinking about enrichment: practical enrichment, which reduces noise; prognostic enrichment, which can increase the event rate; and third is a predictive enrichment to select for responders. And often this is a pharmacogenomic biomarker. And again, if you Google enrichment guidance, you would get the PDF. The third one is companion diagnostic. A companion diagnostic is a test that is essential for the safe and effective use of the therapeutic product that it's co-approved with. And this test can be used to select patients, monitor response, or select dosing, etc. And this guidance describes FDA's policies for approval and labeling of the therapeutic product and the companion diagnostic pair. It discusses about the analytical validity of the test that's used for critical decision making. And the general expectation is the contemporaneous approval of both the therapeutic product and the companion diagnostic. However, this guidance does not describe how to co-develop these products, and that's discussed in the aptly named co-development guidance, which is kind of how to, or the practical aspects of co-developing a companion diagnostic with a therapeutic product. And it describes in general terms the principles that guide the co-development in order to obtain the contemporaneous approval. It discusses some of the regulatory requirements, and it provides considerations for the therapeutic product's clinical trial when an investigation of IVD companion diagnostic is added to that clinical trial. Those were the 4 guidances that I wanted to briefly talk about. The other one that's on the horizon is the complementary diagnostic, the PDL1 example that I had mentioned earlier during the talk. Complementary diagnostic is an in vitro diagnostic that identifies biomarker-defined subset of patients that respond particularly well to a drug, and this aids in the benefit to risk assessment for an individual patient. And unlike the companion diagnostic, a complementary diagnostic is not prerequisite for receiving the therapeutic product. That is, the drug is indicated to an all-comer population, but the test results may suggest who respond better when compared to the all-comer population. So in summary, now genomics is being incorporated into all phases of drug development. Commitment to precision medicine has led to the development of molecularly targeted drugs. Having a strong scientific rationale and having a strong scientific foundation increases the probability of success for a drug. Drug diagnostic pair co-development is possible, and we need to be mindful of the pitfalls or the gaps in the future landscape. And precision medicine is certainly great for patients. It's both exciting and challenging for researchers, regulators, and clinicians. Before I take your questions, I just wanted to quickly switch gears and talk about regulatory research at the FDA. FDA offers many exciting paths to learn about regulatory science, and there are programs that are targeted to different phases of the career. There are programs that are for undergraduate and graduate students. There are fellowship opportunities for postdoctoral fellows, and there are even faculty and senior scientist programs. Again, I don't know if you have access to these slides. I would say if you don't, just Google for FDA studentship fellowship, and you will land at this page. And from there, you will be able to find all the information that you would need. If you're specifically interested in clinical pharmacology fellowship, email ocp@fda.hhs.gov with your questions and inquiries.